Right, so last time we started talking about multiple testing. On Monday, we talked about multiple testing. This is where we stopped. So here, our differential gene expression analysis is one very typical application of multiple testing in bioinformatics. So the reason why it's multiple, we call multiple testing is because we have multiple genes. So each gene needs one test to decide whether we find this gene a DE gene or not. So in the definition of a DE gene, it means that we reject the null hypothesis that the gene has the same true expression value under the two conditions. Okay, so the multiple testing means that we look at m genes in total together and we do m tests. And I wrote m comparisons just because one criterion we need is per comparison error rate, PCER. Here, the comparison is a two condition or two group comparison. We're comparing a gene's expression levels under two conditions and using that to make a decision of rejection of the null hypothesis or not. So this decision table forms the basis of when we discuss the basis for our discussion of multiple testing. So we have the rows as our decision. How many do we reject? How many we don't reject? And the columns as the ground truth, which we don't observe, but we have it hypothetically. How many are true null genes, that are true DE genes? How many are true non-DE genes? Okay. And so M is the number we know, but all these four entries are not directly observed because we don't know the truth. But we do know is the number of rejections total, and that's R, and M minus R is the number of no rejections. And M0 is the number of true DE genes, and M minus M0 is the number of true non-DE genes. So the first criterion is the per comparison error rate, which is basically the average number of false positives. So this one, let me put it here. When the null is true, but we reject it, we call it false positive, FP. When the null is false, but we don't reject it, we call it false negative. Okay, so FP and FN are the two things we want to make small. Sorry, FP is the V, FN is the T. So PCER is basically expectation of V, false positives, on average across all my P comparisons. So this criteria can be upper bounded by changing M to M0 because M must be greater than M0, right? M0 is the number of true negatives. And then this one, expectation of V, can be written as number of true zeros times the type 1 error or significance level. That's what our p-value threshold is. We call this alpha. Typically, alpha is set to 0.05 in practice. So therefore, you can see this PCER is actually upper bounded by alpha. So what this means is that if you set your p-value threshold to 0.05, then this PCER will be bounded by 0.05. So per comparison error rate can directly be controlled with your p-value threshold, in other words. And the second one is more stringent because we may not be happy to just control PCER since this expectation of V, this total number of false positives in expectation, EV can be very big, right? If your M0 number of true non-DE genes, if it is 10,000 with alpha as 0.05, right? You may still have 500 false discoveries, and that can be too many. So to avoid that, a more stringent criteria is called family-wise error rate. That is, we directly control the likelihood or the chance that V is greater or equal than 1. So, but this is so stringent that we have to rely on probability to control it. And this is a typical way of doing an upper bound of the probability of a union. So this is something I explained during the office hour. That is the probability of two events union. That is one of them happens, you have the event happen. And this union probability is less or equal than the probability of their individual events and the sum, right? Sum of individual probabilities. So we're using this as a very loose upper bound because this doesn't depend on any assumptions about the independence or dependence of the genes. So regardless of that, this always holds. And therefore, if we use this upper bound, we just need to set our p-value threshold to be a very small value. That is our target family-wise error rate Q divided by the number of tests. So if Q is set to 0.05, and if the number of tests is 10K, then you can imagine this is very small, right? So it used to be 5 times 10 to the negative 2 for Q. Now it's 10 times now it's 5 times 10 to the negative 6. So that will be a very small threshold. And changing the threshold, p-value threshold, by dividing the target level by the number of tests is known as the Bonferroni correction. So this value or this criterion, family-wise error rate, may be reasonable if your number of tests is not big. For example, if you just have 10 tests, then dividing 0.05 by 10 is still some threshold that's not too small. But in such high throughput genomics, right, with so many tens of thousands of genes, that's not the case. So that motivated us to develop a new criterion, where a new criterion became popular, and that is called false discovery rate. Short as FDR. Okay, so basically, in short, FDR is a new criterion which allows you to set a p-value threshold that is not as small as Bonferroni correction, but not as loose as the no correction case, right? If we just want to control per comparison error rate, we just use the p-value significance level 0.05, and there's no correction. And this means extreme correction, but FDR lies in the middle. So what is the definition of FDR? FDR is defined as the expected value of V over the maximum of R and 1. So going back to the table, it's the V false positive over false positive number over R, total number of rejections. Okay, so which is why it's called discovery rate, false discovery rate, because R is the number of rejections, so the number of discoveries. And we're interested in controlling the proportion of false discoveries among the R discoveries. Now, why do we need to put this maximum between R and 1 in our denominator? The reason is because we don't want to have this dividing by 0 issue. What if we have 0 discoveries? R and V are both 0. Then we have 0 divided by 0. That's not something we can mathematically be valid, right? So we want to avoid that. So this is the false discovery. So compared to this family-wise error rate, you can see this is less stringent because, for example, if I want to control false discovery to 5%, that means if I have 100 discoveries as my R, then only about 5 are expected to be false positive. So that is what we aim to control. And it's not as stringent as this one, because if it's 5 false discoveries, of course, if that satisfies this event that V is larger or equal than 1. So this is something we don't want to tolerate when we control family-wise error rate, but it's something we can tolerate when we talk about FDR control. Okay, so in other words, you can see that when we set a p-value threshold in multiple testing setting, we are essentially following a criteria. Given different criteria, we may set different thresholds. Sorry, I can't read the denominator for FDR. Max R1. Yes, we're taking the maximum of 2 numbers, R and 1. So to control the false discovery rate, how do we do it? So this criterion was proposed in this Benjamini, Hochberg, 1995 paper. It was a statistics paper and probably one of the most widely cited paper because it proposed the FDR criterion, and later FDR has become so popular in biomedical research for setting p-value threshold. So thanks to the author's name, the procedure for controlling FDR, say how to control it under Q. For example, 5%. As they propose a procedure to do that. And this is called Benjamini-Hochberg BH procedure. So what does it do? So it has multiple steps. So first of all, it will rely on the p-values of the m tests. So it will use p1 to pn as the m p-values as the input, and then try to set a threshold, all right? So step 1 in this procedure is to sort or order the p-values from the smallest to the largest. And we use the notation, we put a number in parentheses to mean that it's ordered. So P1 is less or equal than P2 is less or equal than Pm. So this P1 must be one of the P and P values, but we don't know which one it is. Just notation-wise, we just use P parenthesis 1 to mean the smallest P value. Okay, that's what we do. And correspondingly, their null hypothesis is sorted in the same way. Correspondingly, the null hypothesis, we call them H1, H2, HM. This is the corresponding nulls. These are the null hypotheses. And what do we do with that? So step 2 is to do this sequential check. That is, we compare the ith ordered p-value, let me just call that pi, to this changing threshold, which is the q you target times the order I over the total m. So you basically do this comparison from I goes from 1 to up until n. So you started from the smallest one, P1. So basically what it do is that you compare the smallest p-value to Q divided by m. That's your first comparison. And you see this threshold is the same as the Bonferroni correction. Okay. And you compare P2 to Q times m times 2, and P3 to Q divided by m times 3, and so on. So essentially, as your p-value order increases, your threshold increases. So you're not comparing every p-value to this Q over m very small threshold. That's why you are expected to find more discoveries using Benjamini-Hochberg than Bonferroni. So you do this comparison sequentially until it breaks. What it means is that this inequality no longer holds. So you stop whenever it doesn't hold. So in other words, you want to find, in other words, you want to find K such that for all I between 1 and K, we have find the largest, I should say, find the largest K such that for all I smaller or equal to K, we have Pi less or equal than Q over M times I. That's it. And then we reject P1 up to Pk. No hypothesis. No hypothesis. H1 to HK hypothesis. Yeah, so that's what we do. So essentially, we are finding the threshold using all existing p-values. And this is also different from the PCER, per comparison error rate, and family-wise error rates, because for these two criteria, the p-value threshold is set just by our standard, right? PCER is just our significance level alpha, while for family-wise error rate, it is just the target level, say 0.5, 0.05, divided by number of tests. Only for false discovery, the threshold is more like saying data-driven, because we need to see our NP values P1 to Pn to set the threshold. We cannot know the threshold beforehand. Okay, so then you can see why I said this is less conservative than the Bonferroni correction. So let's say that given target level Q, okay, say 5%. Then for PCER, the p-value threshold, or maybe people call it alpha in many cases, PCER just has alpha set to Q, okay? While family-wise error rate, we can set alpha as Q divided by M to control it to Q. While for FDR, we will have alpha, you know, who knows, but it is definitely bigger or equal than Q over M. As long as this one, as long as the first p-value holds. If this inequality holds, then the p-value threshold must be bigger or equal than Q over M. So that's why you are less stringent, because in this case, we are comparing each pi versus Q. And here we are comparing each pi versus Q over M. And here we are comparing the ith order pi versus Q over M times I. So that's why we're comparing making different thresholds. Okay, so why does this BH procedure work, right? Why does this two-step thing, sort p-values and then do the sequential comparison with this increasing threshold? Why can it give us FDR control? So a very, how to say, not rigorous proof, a very short but intuitive proof of this can be seen in the following way. So suppose that, okay, the proof of the BH procedure for FDR control. So informal proof can be think about in this way. So let's do this in the conditioning. We know that R is random, right? Number of rejections, how many null hypotheses we reject, that is a random number, because depending on our data, that number can differ. But one thing is that we can put R in the condition when we talk about this expectation of this ratio, okay? FDR definition, if we do that. So basically, we say conditional on R being K. And K is where we stop, right? The largest K such that this inequality holds for I from 1 to K. Then we stop there, because when we add K plus 1, the inequality breaks and we don't move forward. So we have K rejections in this case. And therefore, if we look at this conditional expectation, V over R given R being K, what is it? So because R has not been in condition, okay, so R can be treated as a constant, K is in the condition, we can just put R as K, and we just have expectation of V. And in this case, since I should say expectation of V, given that R being K, I should write it this way to be exact. And given R being K, it means that we know the p-value threshold is said to be what? is said to be the largest p-value that's rejected, and that's pk. So pk is essentially the p-value threshold we choose, right? We cannot choose larger than that. So in this case, the number of false positives is actually M0, number of true nulls, times the p-value threshold, and that's p this divided by k. Okay, and we also know that this pk still satisfies this inequality. So we have, I just put M0, okay, pk itself will be less or equal than q divided by m times k. Okay, and furthermore, I can make M0 bigger to be m. Okay, and then you see I have all these cancellations, and that is q. So this is the informal proof. It shows that given the conditioning that we make k rejections, this procedure can make the conditional FDR to be less or equal than q. Okay, and then the next step is that we can move the conditioning. But the more exact proof needs some more advanced techniques, which we'll not cover. So I hope this gives you the intuition about why this happens. All right, so, and I think that's it. So basically for multiple testing, you will need the following. So I'll just do a summary. So how to do multiple testing? For each, the first step is for each test, you need to specify what is the null hypothesis, what is the alternative hypothesis. That's the first thing you need. Sorry. Yeah, that's the first thing you need. You need to specify the null hypothesis and alternative hypothesis. And the second thing is that you need to specify, maybe select each test I, okay? And specify test, what is the null and alternative hypothesis for this, for example, gene I. And second, you need to specify the test statistic. The test statistic, let me call that TI for each gene, like gene I. Third, you need to know the null distribution of TI, which is under the null hypothesis. How is the TI distributed? Okay, the fourth one is to obtain a p-value. We call that PI, which is based on the data you have. You calculate the value of the test statistic, compare the value to the null distribution, and use the tail probability as the p-value. And which tail should you use depends on your alternative hypothesis. So this is what we do for individual tests. And the second is that suppose you have the p-values. Given the p-values, P1 to Pn, we calculate in step 1. Then the second step is how to set a threshold. Okay. Set a p-value threshold. And we use 3 ways. So given if you want to just control the PCR, that means no multiple testing correction. And there could be a large number of false discoveries in your result if you do that. And I think a lot of the scientific journals don't accept this because there's nothing you can say about the number of false positives. Second one, family-wise error rate. This is the Bonferroni. And this is very stringent. And some people may say this is conservative. So in the statistical test scenario, when we say conservative, we mean that we are less likely to make rejections, or we stay with the null hypothesis, we don't reject it, and that is considered conservative because only rejecting the null hypothesis is considered to make a discovery. Finally, FDR. And for this one, we have the Benjamini-Hochberg procedure, and it is less stringent than family-wise error rate. All right, these are the 3 different criteria we can use to set p-value, set a p-value threshold. Okay, so one more thing I want to say is related to FDR. Yes, there's a hand.
Yeah, Professor, I had a quick question. You mentioned that currently in the literature, most papers employ FDR. Is there a reason why um, it would be advantageous, or is there a reason why any lab may choose to look at PCER or family-wise error rate, given that, PCER would lead to inflated false positives or many false positives, and then
Yeah,
family-wise error rate is super conservative.
yeah, exactly. Because for PCER, you cannot say anything about how many false positives there could be. They could be very large, especially in DE analysis. If we have, like I said, if you have 10K genes, and if we know the majority of them are true non-DE genes, if that's our belief, then at 5%, at 5% p-value threshold, the PCER is controlled, but you can end up getting 500 false positives. So maybe that's too many for you to afford because you don't want your experimental validation to likely fail. But with family-wise error rate, you may end up getting not much, maybe just 2 or 3 genes, because it's the threshold, this p-value threshold is just too small, and you are not happy, right? You think a lot of the signals are missing there. So when you are being conservative, you are also losing your discovery power because you don't call many genes DE genes. So the FDR became popular because it's in the middle and it can give people a decent number of discoveries to continue from there.
Got it. Thank you.
Thank you. Alpha, there's a question in the chat box. Alpha is the rate of FP, right? Do you mean the rate of false positive? Alpha is the so-called significance level, which is when the null hypothesis holds, what is the probability you rejected? Yes, so it's basically the probability of a false positive, but I have to be a little bit clear that it is a conditional probability. That is, given your null holds, the chance that you rejected by mistake is alpha. So that's why we call the significance level or type of error. Is there a way to control false negative? That's a good question. That is the other side of the question when people compare power. Because power is what? Power is basically 1 minus false negative rate. So that is false power means if the alternative hypothesis holds, what is the probability you make rejection? Okay, so that is 1 minus false negative rate because false negative rate is when the alternative holds, you don't reject it as false negative. So therefore, we compare methods only under the same FDR or p-value threshold. So we have to do that. So it's a fair comparison because it's always a trade-off. I can achieve perfect power. That is, I discover everything, but I just cannot control anything about false positive. So therefore, when we compare power, it only makes sense when the FDR is at the same target level. Does that make sense? Yeah. So I would say for any method, any test statistic, what you can do to set a p-value threshold is to control the type 1 error, the false positive thing. And then if you have multiple choices for your methods, you can compare their power and say which one is more powerful. Okay, and something maybe I can say a little bit about this, since we already have this table, are some terms we often encounter in practice. So these are the terms related to, I will see, just introduce some terminologies. All right, so we have introduced type 1 error, right? Or type 1 error, it is the false positive rate. It is what? It is the number of, I'll just use the words instead of the notation, not to confuse you. It is a number of false positives over M0, it should be, okay? Or in my previous notation is the V over M0, if you still remember that. That's type 1 error. And type 2 error is the false negative rate. It's the expectation of false negative over the true positives, M minus M0. So if you remember my notation here, this Fn is T. But please note that in these two expectations, the denominators are constants, so I can get them out of expectation. So it's basically E of V over M0. They're not random. E of T over M minus M0. Okay. And what else? I said power is 1 minus type 2 error. Okay, that's our definition of power. And here the type 2 error and power are defined by pooling all M tests together. So this is like an aggregate metric for all M tests. But just bear in mind that we can still use the term type 1 error, type 2 error power for a single test. When we're doing that, then we're not looking at M genes together, just one particular gene. We can still talk about how likely, if the gene is a true non-DE gene, we mistaken it as a DE gene, then that is type 1 error probability. So we can also say that for a single gene. But here the terms I introduced are for all M tests jointly. Okay, and furthermore, there are something you may also encounter in your data analysis. One is called ROC. It's called receiver operating characteristic. ROC curve. So what this one is plotting is actually on the x-axis, you plot false positive rate. Okay, and in the y-axis, you plot what? You plot when false positive rate is 0, then you're making no discoveries and your power is 0. You plot the power. So essentially, the curve will have dots following your different, say, p-value thresholds for making your decision, and you calculate the two corresponding values. So you will definitely have the curve go through 0, 0, and 1, 1. So that makes sense, right? Because when it's 0 false positive rate, that means I make no discoveries. And if I make no discoveries, I have no power. On the other hand, if I recall every gene as a DE gene, I have perfect power, but then I may have a very, very high false positive rate. So in that case, the false positive rate is 1 because all the null cases are called as discoveries. So that's why when we compare ROC curve, we are having one curve per test statistic or per procedure or per method. And the points along the curve corresponds to different thresholds you set on the method's output, for example, p-values, because at different p-value thresholds, we will have different trade-off between FPR and power. And when we compare the methods ROC, we will go with the method that's right here. So if a method has like a sharp rise and then go flat, it means it's optimal. Basically, it means that as long as you can tolerate a little bit of FPR, power becomes very high, and then you can find a lot of discoveries. So it's always related to how we can distinguish signal to noise. And one idea to think about this is if you think about your test statistic distribution. Okay, let me just draw this. So this one, if I mean, is the distribution of T under null, and this is the test statistic distribution under alternative. Essentially, our decision threshold, if we cut it here, okay, then this will show us that to the right of this T under the null distribution, this area would be my type 1 error. Because that's when the null hypothesis holds, but we make rejection. And to the left of it, this is under the alternative, even though we should make rejection. But when it's less than T, we don't make rejection. This is our type 2 error. So this ROC can be drawn as you vary your T. You can vary your T to be very small. In this case, your false positive rate will be high, but your power will be very low. And if you go to the other end, your false positive rate will be very low, but your false positive rate will be very low, but power will also be very low. So basically, you can see that by changing this T, you will be able to get this curve. And the two distributions overlap. How much they overlap would determine how well this ROC curve would be like. Because you can imagine if we have them perfectly separated, then if I draw a threshold in the middle, then I can have no false positives and perfect power. So different test statistics, some being more powerful than others, will actually be reflected in how much their distributions overlap under the two hypotheses. Does that make sense? Okay. And this is the ROC curve. Another thing we usually do is called the precision recall curve. And people sometimes call this PRC. Okay, precision is closely related to FDR. Basically, precision is very similar to 1 minus FDR. Even though I'm ignoring a little bit formality here because I'm ignoring that FDR is an expected ratio, but precision is a ratio that's without expectation. But conceptually, they are related in this way because the precision can be said as, if we follow FDR expectation, is expectation of my true discoveries among my discoveries. Okay, so it's S over R. That's my precision. And recall is another way of saying power. So they are the same thing. So for recall, it is among my true positives, how many do I correctly discover? So in other words, for recall, it is expectation of S over N minus M0. The true positives, how many do I discover? So precision recall curve is also used as an parallel to ROC curve, especially in classification problems where you want to predict whether one individual has a disease or not, right? Remember, I mentioned this distinction between hypothesis testing and binary classification in the first class. But in classification setting, ROC and precision recall curves are used a lot because people want to say, yeah, so for the precision recall, something that make it more desirable than ROC is when the two classes, the positive class and negative class, are very imbalanced, which means that you have one class that clearly dominates. So one class has 90% of chance and the other class has only 10% of chance. So why is the ROC in that case not so meaningful? Because you can think about in this case, right? If you make a mistake, so in this case, if you use ROC, then because the minor class is just too small, then you would only have a very small difference in distinguishing two methods. Because you could say, if I just predict everything to be my positive class, then my overall accuracy, which is a weighted sum of type 1, type 2 error, can be as high as 90%, which seems to be quite good, even though I completely ignored my minor class. But that is something you will not see in the ROC pattern because the denominator for the minority class, the total number, like M0, for example, is just too small. Then a little bit change in your decision threshold will have a sharp rise in your curve. The precision recall curve is different because you can see in precision, your denominator is not true negatives, but it is your discoveries. So therefore, how many you discover can be accounted for. While this is not in the type 1, type 2 error, both denominators are the truth. The true positives, true negatives in your ground truth. But here precision, like FDR, has the denominator as your number of discoveries. So therefore, if you discover less or if you discover more, that has to be accounted for. So there are some empirical evidence. I think if you just Google, when should I use precision recall curve, then you will see that people generally recommend using that if you have this imbalance scenario in the binary classification setting. So basically, both curves are trying to summarize the accuracy of a classification algorithm and showing the trade-off as you change your threshold between true positive and true negative. Even though I want to say that our our multiple testing problem is not the same as binary classification conceptually, but the way we define metrics, how do we define a metric based on the results of the M tests, that is considered similar. So we also have this decision table based on our decision, how many rejections, how many not rejected versus ground truth. So for that type of two-way decision table, they are similar, but conceptually, multiple testing and binary classification are two different things because multiple testing, each test is about one feature, one variable, while binary classification, each decision is made for one observation. That's something I said in the first class. Okay, any questions before we move forward? All right, if not, then I think the next thing I hope to introduce to you is what I said earlier in the class about, I think we said that last time, negative binomial distribution. And many people call this MB distribution. Okay, why do we want to introduce this? Because it is a distribution that is commonly used to describe counts. It is for count data. So a very typical example where negative binomial can be a good distribution is for single cell RNA-seq data. Single cell RNA-seq data. And the data, I would say, to briefly introduce it, if you do the sequencing and you run the software packages for processing the raw reads, you would end up getting a count matrix. So depending on how you write it, but I think in single-cell RNA-seq data, there are two popular packages, one called Surat for R, and for Python, there's a package called ScanPy. So you have these two packages that are popular. And in Seurat, I can say the data is stored as a matrix and called Seurat object. But this object is actually the orientation is different from what we typically say in statistics, which is we typically have the cells as our rows and we have genes as our columns. But in Seurat, if you look at it, genes are in the rows, cells as columns. So therefore, it's very important that you first make sure which are which. And the reason I prefer to have cells as rows because I think it makes more sense to consider cells as observations, a sample from a cell population, but by treating genes as variables or features. So that follows the convention I advocate in the first class. We put observations as rows, we put variables or features as columns. Okay, so we have a count matrix. And in this case, if I use Xij to denote a count. So basically, once we do data pre-processing, Xij will be the count of gene J in cell I. Okay, so for single cell data, one important task is to identify cell types from the cells. So how the experiment works is that when you measure those cells and gene expressions within, you dissociate cells in the tissue, making them floating. And then you use the protocol to hopefully wrap each cell into a droplet. And so every cell become isolated, and then you can label the RNA in each cell. And then you load the sample into a sequencing machine to do the sequencing RNA-seq. But since you have labeled the RNA of each cell, the RNA will carry the so-called cell barcode. Then once you obtain the sequencing reads, based on the barcode, you can distribute them to your cells. So you know how many sequencing reads are coming from this cell mapped to this gene. That's how we get this information. And one more thing I want to say about single-cell RNA-seq data is the so-called UMI, unique molecular identifier. Maybe unique molecule identifier. So what this means is you also label the RNA molecules before you do the PCR amplification before sequencing. PCR is commonly performed before you do sequencing since it amplifies the signals. But PCR will also create identical copies of the same RNA. So UMI, since it's labeled before PCR, can help you remove PCR amplification bias. Then you can just count the number of unique RNA molecules of gene J in cell I. So for here, since most cutting edge experimental protocols use UMI, we will stick with UMI as our count. So it's the UMI count. So one question regarding single cell data analysis is how do we model the data? And for now, since I haven't introduced this technology for data analysis in depth, I will say that maybe we could just assume, you can just assume what I told you makes sense. That is, given a cell type, suppose that we can say cell type with n cells, then we can assume that x1j to xnj, the n counts of the n cells for gene J independently follows, IID follows a negative binomial distribution with a mean parameter mu j and a dispersion parameter sigma j. So, or maybe not sigma, phi j. Okay, that's more common. So I'm just saying that in single cell data modeling, it's very common to assume for a given gene, the counts of cells of the same type follows one negative binomial distribution with its mean and dispersion parameters. So that gave me the motivation to introduce negative binomial distribution and its basics. So what is negative binomial? We have to start with Poisson. So let's first look at Poisson as the basis. So Poisson is the most commonly used distribution for counts, right? So it's actually, if you look at the history of Poisson, it is about given a time interval, you wonder how many times an event would happen. For example, if I open a restaurant, given a time interval, how many customers can I expect to during this same time interval? So if I set a time to be 1 hour, then I can collect data every hour, and then I just plot the distribution. So Poisson as a count distribution has a very interesting property. So if we have x1, xij, let's just say, follows Poisson distribution with mean mu j only. Again, here I goes from 1 to n to represent the n cells. Then Poisson has a very important property, that is its mean is equal to its variance. Both are equal to the Poisson parameter mu j, because Poisson is a one parameter distribution, a single parameter. Okay, so this is very important. But if you have real data, you have a bunch of counts, then you can compute the average. What if I compute the average, and so for the expectation, my average to approximate expectation is the average of xij, right? I from 1 to n. And I can call this xj bar, the average of gene bar across my cells. What if this is obviously smaller than the variance of this set of data? And for variance, if I compute it from my data, it's 1 over n minus 1. I from 1 to n, xij minus xj bar squared. So if I have this, then clearly maybe the assumption of Poisson that mean is equal to variance may not hold. Then what can we do with this scenario? So negative binomial was proposed to address the so-called over-dispersion issue of Poisson. So over-dispersion is called here, which means that the variance is bigger. The data spread is bigger than what we expect. So the first derivation of negative binomial is a so-called gamma Poisson hierarchical model. This is the first formulation, gamma Poisson hierarchical model. So basically, what we're doing here is we basically change xij following Poisson, not with the common mu j, because you see mu j doesn't depend on I. It's a gene-specific parameter, but it's the same for all cells. What we did is by changing it to be mu ij. So you see that every count xij has its own Poisson mu ij. Okay, so every cell I for gene j can have its true level, and these true levels mij can be different for different i's. So the gamma is to describe this distribution of mu ij. So we can add one layer by saying mu ij as positive real values follow a gamma distribution, whose parameters are only depending on cell type J, but not cell I. No, sorry, only depending on gene J, my bad, not cell I. So we can call them alpha J, beta J. The reason is that gamma is a two-parameter distribution. But actually, I can tell you that we don't need 3 parameters in MB. MB has only 2 parameters, so we must reduce 1 parameter. And what is that? So this is based on what we expect for the negative binomial distribution. So in other words, we want to have expectation of xij to be equal to what? to be equal to, or maybe I think this is not way to write it, my bad, just bear with me. Probably I should change it to this way. Yeah, instead of saying Poisson mu ij, I stick with mu j times some constant phi ij. Okay, this is easier. And I let phi ij follow gamma. So basically, phi j is a multiplier to mu j to create the Poisson parameter, the Poisson mean to be not exactly mu j, but with some small disturbance or perturbation around it. So therefore, we can account for the overdispersion. Why does the observed variance bigger than the mean? That's the thing. So, but we still want to have expectation of xij equal to mu j. So to achieve that, then as phi ij is random following gamma, mu j is fixed. So we can show that we need to have expectation of phi ij equals 1. So the dispersion, or not dispersion, the disturbance, perturbation, phi ij perturbation factor should have expectation 1. And this will give me that gamma and beta must be equal. And this can reduce one parameter for me. Okay, so this is one way to derive the negative binomial distribution. So basically, following, in other words, if I have phi ij follows gamma, just one parameter alpha j, and xij follows negative binomial distribution with mean mu j times phi ij. In this setup, in this two-layer hierarchical model, I can show that xij follows, sorry, my bad, Poisson, bear with me, sorry. Xij given phi ij follows Poisson mu j times phi ij. Yeah, with this, then I can say, putting them together, the marginal distribution, that is, we integrate our phi ij, xij will follow negative binomial with two parameters. And the first parameter is just mu j. And the second parameter, depending on the dispersion, is something, maybe I can write it out, but it's basically mu j plus mu j squared times, I think it is alpha j, but I might be wrong, this one. So I'm just saying that the variance of the negative binomial is bigger than the mean since we have this additional positive term that add to it. So this can explain why negative binomial has a bigger variance than its mean. So the gamma Poisson derivation is the first way to define what is a negative binomial distribution. But another way you might have learned before is in a probability class where you do the coin tossing. That's another way to define negative binomial. Second way, in the coin tossing example, which is what we define Bernoulli and binomial, right? So basically, in this case, if you want to say x follows negative binomial with also two parameters, but the two parameters mean differently, you can say R and P. R is an integer. Basically, R is the number of failures or number of tails in your coin tossing, and P is just like Bernoulli, is the probability of success, the chance of getting one head. Okay, and X is what? X is the number of successes before, or not before, until R failures. So basically, you keep tossing the coin and you stop until you see R failures, and then you go back to trace and count how many successes are there. Okay, so in this case, we can write out the probability for X taking a value, let's call it K, based on this coin tossing mechanism in a probability calculation. So in this experiment, right, what it means is when I stop, it must be when I see R failures. So the place where I stop, it must be a failure. But how many did we toss? How many times did we toss before the Rth failure? We have tossed K plus R minus 1. That's the number of tosses we have done. And out of those, I know K are successes. That's my X. So therefore, there are so many possibilities of choosing where the K successes are. And for each success, the probability is P, so P to the power K. For each of non-success is 1 minus P, and that's R minus 1 times the final one is a failure, so 1 minus P. So that's how we do it. And the reason we have K plus R minus 1 as a total is because we know the final one, it must be a failure. So there's no randomness there. So using this as the so-called probability mass function for this X as a count discrete random variable, we can compute its expectation. So in this case, expectation of X can be computed as P times R over 1 minus P. And the variance of X is just P times R over 1 minus P squared. And again, you can see because 1 minus P is less than 1, so 1 over 1 minus P is bigger than 1. So this is essentially equal to expectation of X equal to this times 1 over 1 minus P. And of course, this is bigger than expectation of X. So this also shows that the negative binomial has its variance bigger than its mean. Okay, so if I want to find some correspondence between the two formulation, I would say if I want to write it as MB mu sigma squared, mean, let's just say mean variance versus MBRP. Okay, this is the number of failures. This is the success probability. These two ways to parameterize the same distribution is called reparameterization because we just need two parameters, but there's one-to-one correspondence between the two sets of parameters. So they actually specify the same distribution. So essentially, if I want to find correspondence, we can say that P can be written as 1 minus mu over sigma squared, and R can be written as mu squared over sigma squared minus mu. So you can define R and P based on mu and sigma squared, and vice versa. So you can go from P and R and go back to sigma squared. Okay, so that's negative binomial. And as a two-parameter distribution, you may know there's a famous one. There's a normal or Gaussian distribution that's also two parameters, mean and variance. But something I can say is that if you want to do the maximum likelihood estimation of parameters for Gaussian, for normal, it's easy. We know the closed form formula for how to compute the average as the maximum likelihood estimate for the Gaussian mean, and that 1 over n minus 1 variance from our data, 1 over n variance as MLE for the variance. But for negative binomial, there are no easy closed form for fitting it. So we need to do it numerically. So something we should be careful about is that when we do negative binomial parameter estimation, there may be some instability. And this instability can come from the numerical calculation because, as I said, you have to estimate parameters numerically without a closed form. Okay, so any questions so far regarding negative binomial? And I think I want to talk about this because in the previous, when I talk about the differential expression analysis, I said D seek to edge R, they both use negative binomial as the assumption. And there's a question. So going over MB and gamma hierarchy is a way of characterizing count data. So negative binomial is for count data. So this gamma Poisson hierarchical model is a nice way to interpret negative binomial, or it's one way to derive negative binomial, because we are using Poisson as the count generation. But this Poisson parameter, we allow it to differ from cell to cell. So if you think about the Poisson parameter as the true expression level of gene J in cell I, then this true expression level, even among the cells of the same type, they are not identical. And that difference, which we call biological variation, can be captured by gamma. And this Poisson is what we capture by sequencing, what we use to describe the sequencing process. That is, suppose gamma give you the biological variation and you have one true value for gene J in cell I. Then, but you don't observe that. You don't directly observe the true value. The count data you observe is by the sequencing process. And sequencing process can be modeled as a Poisson distribution. That's what I want to say. Okay, so previously I would go over the negative binomial parameter estimation used in the first version of DE-seq. So DE-seq and DE-seq2, if you are interested, you check their paper, they use, even though they both assume negative binomial, but their test statistic and its null distribution derivation are different. So therefore, I think probably for this time, this class, I will not go into that DE-seq framework, but probably I should directly, just stop here saying that negative binomial, even though it's a very nice way to model count data, but we must bear in mind that first of all, check how well it describes our data. Like if you have bulk RNA-seq data, the samples are from diverse patients, not replicates, the negative binomial may not fit well. So the assumption breaks down. Or even if the assumption seems to hold, when you fit negative binomial distribution to estimate the two parameters, we should be a little bit careful about whether the parameter estimates are stable or not. That's something we should also consider. All right, so I think we should probably move on to our next topic. So I think that's what I want to say about the tests. The next topic is about clustering. So I hope to talk about clustering. But before we do that, I hope to share with you this thing from the slides, which is the SURAT for single cell data analysis pipeline. So let me pull that up. And so we can get a sense about how the whole analysis is done. Let's see. Drag it to this site. This one. All right, so this is what I want to show you before we continue with the clustering. So as I said, so the single cell data, you see here, it represents genes as the rows, cells as the columns. So clustering is commonly used in single cell data analysis to identify potential cell types. The reason is that when you sequence the cells, you don't record their cell type information, because single cell RNA-seq is not measuring the morphology or cell surface proteins. So therefore, the typical ways of characterizing cell types is not available in the data, in the count data. So unsupervised clustering, which means that you find similar cells as a cluster, is a way to hopefully help you to decide whether this cluster can be called a cell type. So in this case, you actually annotated 3 cell types of your cells. So let's quickly go here. This is the SURAT steps I want to show you. So first of all, you set up the SURAT object. That's just data preprocessing and standard preprocessing workflow. So this means that you want to remove the bad quality genes and bad quality cells and normalizing data. identifying highly variable features, scaling the data. So these are all data transformation. You're changing your values changing the values in your matrix here. And then linear dimension reduction, which means that you want to reduce the number of dimensions from the number of genes to a fewer dimensions. And then clustering. So this is where Seurat performs clustering. So the clustering of Seurat is not done on the original data. So it's not directly, going back here, it's not directly clustering the columns based on the genes as rows, but they perform extensive data transformation before doing clustering. And I will explain why that is the case. And then they do nonlinear dimension reduction, UMAP t-SNE for visualizing. And then they find differential express features. And then they find the cell types for, so basically the differential express features are what are the genes highly expressed in each cluster. And then they annotate the clusters as cell types. So this is the whole thing. And so far, what we covered is that we focus on the differentially expressed features because it's a statistical testing step. But there are many things we should also consider. So that's why I'm thinking probably I can go with talking about clustering first, but then we will go back to say normalization. What does that mean? Because for single cell data normalization, there are specific considerations for this data type. And then we will talk about dimension reduction, which is also unsupervised. If you have any suggestions, you can let me know. Then how can we integrate the steps or how can we integrate this single cell data analysis steps and the statistical materials we want to cover in a seamless way? Do you have to pseudo bulk by cell type and donor before finding DE features? That's a very good question, but that's a different problem. So for this single cell data analysis pipeline, it's for annotating cells into cell types. So that's why we do clustering and then give every cluster label. What you are talking about is the multi-patient single cell data where the cells have been annotated already with types. And then you use the cell type label. From there, probably you can say, given the same cell type, maybe B cells, how different is it between diseased patients and undiseased patients? That's a different question, right? So here you assume cell types are known, but this is for annotating cell type because you see the last step is to assign cell type identity to clusters. I hope that answered your question. All right, so I expect to assign the first homework by actually today or tomorrow, and probably will give you 3 weeks for doing this homework. So because we have this fire situation and we hope to give everybody enough time to work on the problems. And next Monday is a holiday, so we won't have a class on next Monday. But on next Wednesday, I will need to be out of town because I had a seminar scheduled on that day. But what my plan to do is that I will record a lecture beforehand. And if we're doing this in person back to campus, I will ask a student of mine to help play the video and answer your questions. And I can also answer your questions after I return at the office hour. Otherwise, if it's still a Zoom lecture, then it will be on the Zoom. So same Zoom link as this time. But if you have any questions, just let me know beforehand. And because of the holiday, I think for next office hour, I'll just change it to questions by email. If you have questions, just send me an email on Monday. Yeah, thank you very much. Yeah, thank you.
Thank you.
Thank you. Bye-bye. Bye.